Good morning, students. Today you see the topic Neisseria Gonorrhoeae. Introduction. Gonorrhea is an ancient venereal disease caused by Neisseria gonorrhoeae. So it is characterized by acute urethritis infection with a mucous discharges, infected prostate glands, seminal vesicles, epididymitis, structure formation, water gland perineum in case of male patient. In female vulva, genitals and in a puberty glands, urethritis, the infections of Bartholin glands, endometrium of a fallopian tubes, that means salp sitting extra. So the name gonorrhea means flow of seed, was first employed by Galen in 130 AD. The gonococcus was first described in gonorrheal pus by Neisser in 1879. So hence the genus Neisseria in 1885 cultured the gonococci and provided its pathogenicity. Microbiology. Neisseria gonorrhoeae is a diplococcus with the adjacent side concave. It shows a difficult reniform or a bear-shaped suture formation. So it is a intracellular in polymorph in urethral pus. They are non-motile, non-capsulated, non-sporing and gram-negative diplococci. They possess pili on their surface which help the diplococci in adhesion to mucosal surfaces and promote virulence by inhibiting phagocytosis. So the pili also help in accumulations of RBC. Cultural characteristics. Gonococci are more difficult to grow than meningococci. So they are aerobic and facultative anaerobic organism. They require 35 degrees Celsius to 36 degrees Celsius temperature and 7.2 to 7.6 pH for their best growth. And 5 to 10% carbon dioxide is essential for their growth. They grow well on chocolate agar medium and Muller-Hinton agar medium. And selective medium used for its growth is Thayer-Martin medium. It contains vancomycin, colicins and niacins and inhibit the most contaminations including the non-pathogenic Neisseriasis. So the colonies are small, round, translucent, convex, slightly amalgamated with a finely granule surface and lobal margin. The colonies are soft and easy emulsifiable structure formation. So biochemical characteristics. They ferment glucose producing acid but no gas formation. They do not ferment maltose. So the gonococci resemble the meningococci except in the fermentations of maltose. They are catalase and oxidase positive. When 1% solutions of oxidase reagents, tetramethyl paraphenylamine diamine hydrochloride. So is poured in the culture media, the Neisseria colonies turn the deep purple color formation. This reaction help to identify the Neissers. So the indole and HTS production is negative and nitrate are not reduced, nitrate reductions of negative reactions. Antigenic properties. So the gonococci are antigenically heterogeneous. 3 antigenic structures are found in gonococci. The first one pili, second one outer membranes protein and protein 1, 3 and 2, outer membrane lipopolysaccharide endotoxins. And first one pili are a hair-like structure formation. The several micrometers long composed of a peptide subunit and prions. They have consistent and variable region. So they act as the virulent factors by helping in attachment of the host cells by inhibiting phagocytosis. So they shows the antigenic variation. Outer membranes protein are different types. Protein 1, 2 and 3 are important among them. The lipopolysaccharide means exotoxins are responsible for toxicity in gonococcal infection. Resistant. Gonococci are very delicate organism readily killed by the heating, drying and antiseptics. It cannot survive outside the body. It dies in 1 to 2 hours in the exudates. The cultures die in 3 to 4 days, but in slant they are surviving 35 degrees Celsius if kept under the sterile paraffin oil. The culture can be stored for the yeast at minus 70 degrees Celsius or lyophilized. It is formerly highly susceptible to sulfonamide, penicillin and other antibiotics, but now developed the resistance to many antibiotics, especially penicillin due to penicillinase production. Pathogenesis. So gonococci is a venereal disease occurred by the sexual contact. The first steps in infection is adhesion of a gonococci to the urethral or other mucous surfaces. So the pili help in adhesion. Adhesion is rapid and firm, so there is no production after expose. So gonococci penetrate through the intracellular space and reach the submethelial connective tissues within 3 days. Then incubation period is 2 to 8 days. In man, the disease start and acute the urethritis with the mucofloragen discharges having gonococcal infection. So infection extended to the prostate glands, the seminal vesicles and epididymitis. The chronic urethritis leads to the stricture formation. If it is spread to peripheral tissues, it causes the abscess and water perineum. Next one, in women, the infection involved the urethra and cervix urethritis due to acid pH and resistant stratified squamous muscle epithelium, and adult in general and are not affected. So in pupillary glands and vulvaritis occurs. In women, infection extended to the birth-zone glands, endometrium and fallopian tubes, and soliciting may lead to sterility. Asymptomatic carriers are more in women than men in both sexes, and proctitis and conjunctivitis, arthritis, ulcerative endocarditis, rarely meningitis may occurs. Lab diagnosis. So diagnosis is easily in acute stages. Then in chronic cases, samples are collected from urethral discharge in both sexes, and in women, cervical swabs are also used. First one, the microscopic examination. The smear is prepared from the urethral discharges. It is a gram stained and observed. An intracellular gram negative diplococci give the presumptive evidence in men. But is woman normal flora may have a similar morphology, so the fluorescent antibiotics techniques is used. The fluorescent antibiotic techniques is more sensitive to and specific than diagnosis by the microscopy. Cultural method. The specimens are inoculated on a chocolate agar medium or a Muller-Hinton agar and incubated at 35 degrees Celsius to 37 degrees Celsius under 5 to 10% of carbon dioxide. To isolate the gonococci, culture should be followed by microscopy. So epidemiology. So gonorrhea is a exclusively human disease. The only sources of infection is human patient or carrier. The mode of infection is exclusively venereal disease. So prophylaxis. So gonorrhea can be controlled by the earlier directions of cases, healthy education and safe sexual conduct, and vaccination has no place in the prophylaxis. Treatment. So sulfonamide, penicillins are effective antibiotic. When the strains develop resistance, cefotaxime, erythromycin, tetracycline, ciprofloxacin are useful. Thank you.